All right, get started here. Um, so again, we're going to go into biotechnology and DNA technology. We've talked a little bit about this already. Let me get my camera on. In the last chapter, when we talked about DNA and bacterial genetics, we're going to get a little further into it, and you may recognize some of this stuff because you may have done a project over that. Now, on Thursday, I am going to present all your projects to you guys, right? And let you watch everyone's submission. And that way we can learn from each other about some of the biotechnology that is out there. I don't know if I'll finish this completely today, but we'll work on that. Okay, I got you. So anyway, we'll watch those on Thursday, and we'll probably may even bleed over into the next week after spring break. We'll watch the rest of them. But continue working on your disease project. Again, there's a sign up sheet for that. Make sure that I approve what you're doing. I'll try to check that tomorrow. I haven't looked at it yet to see if anybody submitted anything, but if you haven't, go ahead and do that. Is anybody else having problems with the audio? Uh-oh. Nikita, you can hear? All right. Let me jot down the roll real fast, and then I can log back out, log back in. I guarantee it's something to do with my phone. Yeah, the sound's on. It's on. It should be working. Um, I'm wondering if my phone's not interfering with it where I signed in on it. Let me exit out of that. And all right, let me get rolling. I'm gonna log back out, log back in. Is it set for Friday? I thought it was. Yes, it actually is. I'll move that to Sunday. I usually make everything due on Sunday, so I'll do that. But let me log back out and come back in and see if I can get the sound fixed. And yes, you will get a study guide tomorrow. Um, I've had a busy weekend with moving and stuff, so I hadn't had a chance to look at that. All right, give me one second. Let me get roll and then. Okay, well then, if y'all will do that, then since everybody else is able to hear, then I'll just leave it on, and I'll come back to it. Let's see. All right, so since everybody can hear, I'll do roll when we get done. So anyway, we'll get started with biotechnology and DNA technology. Again, some of this will be repeat, some of it you may recognize from your own projects, but we'll get into this and see how far we can get along with it. Hopefully we can get most of the way done. So this is just a brief video to kind of introduce you to biotechnology. And while we're watching this, I can take roll. Uh-oh. That's not good. All right, why is my video not working? All right, let me check on something else. Let me see if this video is going to come up. All right, let me close this out and see if I can come back in. I have all kind of issues today. Oh, I bet that's it. Here we go. That's what it was. All right. Yeah, all right. Our videos are working. All right, so this is going to go through biotechnology and just kind of give you an overview of all of that. Yeah, biotechnology is due tonight at midnight. All right. Make sure you get that in because I'm going to try to grade those tomorrow. And then again, we'll present those on Thursday. So anyway, I'm going to cut my mic off and my camera off so it won't interfere. But this is just going to give you a brief overview of biotechnology over the years.
As some scientists worked to control life at the scale of global agriculture, others worked in a different direction. The mid-1900s was a period of re-examination of one of our big questions. What exactly is life? Let's talk about DNA and biotech. Although the story is complex, it's often simplified to one big discovery of DNA made in 1953 by two dudes who won Nobels. Turns out there were also other people involved. By the 1940s, researchers knew that the cell nucleus contained thread-shaped structures called chromosomes that played a critical role in cell division. Chromosomes seemed to be made of a mixture of protein and other stuff. And this other key stuff was a molecule made out of carbon, hydrogen, nitrogen, and phosphorus. This was deoxyribonucleic acid, or DNA. Isolated DNA looks kind of like a white powder, but no one knew DNA's structure. A molecule structure, the way it fits together, tells us about how it works and maybe how to redesign it. In 1944, Austrian physicist Erwin Schrödinger, yeah, the cat guy, published a short book called What is Life, reviewing this deceptively simple question. Scientists knew that there must be a unit of heredity, the gene that must be part of the chromosomes. Schrödinger examined the laws of physics, determining that the gene must be very small, only a few thousand atoms in size. It must vary, yet it must be orderly and not give rise to too many mutations. So Schrödinger threw down the challenge. How does this gene physically encode the information that defines life? He argued that this was among the most interesting questions facing science, and he suggested that one of the people best poised to answer it was biophysicist Max Delbruck. Delbruck ran a loosely organized network of researchers at Cold Spring Harbor Laboratory, Caltech, and elsewhere called the Phage Group. The group worked with viruses that parasite bacteria called bacteriophages. Viruses are just nucleic acids and little protein robot bodies. The Phage Group did important work on how life works at a small scale, using radioactive tracers inside viruses. But even they couldn't tell if it was the DNA part or the protein part of the virus that took over the bacteria. And no one could explain how either physically encoded information. So by 1950, the pressure to understand DNA was on, even though not everyone was convinced that DNA was the physical substrate of heredity at all. Despite this uncertainty, scientists set out to win this race. The most famous was American chemist Linus Pauling, who went on to join the short list of people with 2 Nobel Prizes. Pauling was an obvious choice because in 1951, he characterized the alpha helix structure of common proteins. He used an empirical approach, X-ray crystallography. X-rays, which have wavelengths much smaller than visible light, pierce molecules, then scatter, making a diffraction pattern that reveals information about the molecule's shape. Crystallography is an incredibly finicky technique, but Pauling correctly showed how common proteins fold up into elegant little spirals. He then decided to tackle DNA, guessing incorrectly that it was made up of 3 helices. Also in the race was James Watson, a brilliant, young, and brash American biochemist. Brash, of course, being the historian's euphemism for sexist jerk. He was a member of the Phage Group and a fan of Schrödinger's What is Life. Watson traveled to the University of Cambridge's Cavendish Laboratory. There he partnered with English biophysicist Francis Crick, who became one of the great theorists of modern biology. Watson and Crick's approach was modeling DNA, asking which atoms went where based on the laws of chemistry and physics. Now, if you've read Watson's best-selling autobiography, The Double Helix, you'd think he and Crick did the heavy lifting in discovering the structure of DNA. You wouldn't know that Harvard University Press refused to publish his book because of its potentially libelous characterization of their collaborators. Thought bubble? Show us another side of the story. Watson cast English chemist Rosalind Franklin as the villain. Franklin worked at King's College London, not the Cavendish, and she was Jewish, and she was also a woman. She also went to a talk by Watson and Crick and tore apart their suggested model of DNA. The head of the Cavendish was humiliated, forbidding them from more DNA modeling. You see, Franklin was a leading expert in X-ray crystallography. Her photographs had shown that there were 2 forms of DNA, A, which is dry and crystalline, and B, which is wet, how DNA looks in living cells. This discovery was a fundamental step in understanding DNA. We now know that there is a 3rd form, zDNA. Then in 1952, Franklin made one of the most famous photographs in science, Photo 51. It shows a clear X pattern, the signature of a helix or spiral stair shape. But Franklin didn't know that the deputy director of her lab, Maurice Wilkins, was secretly passing her notes and images to Watson and Crick. The rest became history. In 1953, working on their model, reviewing facts about the 4 nucleic acids in DNA, or bases, and looking at Franklin's images, Watson and Crick realized DNA must be a double helix, and that the bases must be paired so that the A's equal the T's and the G's match the C's. The zipper shape of the double helix allows DNA to transmit information from generation to generation with few copying errors. A cellular machine unzips the staircase down the middle and figures out one half of a base pair by looking at the other. If one base is an A, it must connect to a T. Simple. Watson and Crick invited Franklin to Cambridge to review their work. She immediately acknowledged that it was correct. She just didn't know how much they had relied on her own work. Thanks, Bubble. After publishing their model and the data backing it up, Watson and Crick became scientific celebrities. Franklin, however, died prematurely of cancer, likely due to her work with X-rays, and the Nobel Prize is not awarded posthumously. So in 1962, Watson, Crick, and Wilkins shared the Nobel without acknowledging the debt they owed to Franklin. But in part because Watson described Franklin so horribly in his book, he called Franklin Wilkins' assistant, historians went back and researched her life, writing her back into the role of protagonist in the story of DNA. So a scientific object like DNA is assembled out of other scientific objects, such as X-ray images, textbooks, and three-dimensional models of tin and cardboard, but also erroneous ideas such as Pauling's triple helix, as well as relationships and competitive drives for fame. With DNA revealed, life itself could theoretically now be not only read, but programmed. Remember, this was around the same time as the birth of computing. So DNA became a machine language program to make RNA, which became an assembly language program for making proteins, which are what life is made of. This process was thought to be quite computer-like, moving only in one direction, from DNA to RNA to proteins. This rule, first expressed by Crick, is the central dogma of genetics. We now know it's more complicated, but the essential idea is useful. The question after 1953 was another how. About the genetic code. DNA has 4 nucleic acid letters, A, T, G, and C, with a U instead of a T, and RNA. But how do these code for the 20 amino acid letters of the proteins that we're made out of? Some of the DNA discoveries went back to the theoretical drawing board. In 1954, Watson and Soviet-American physicist George Gamow founded the RNA Tie Club to figure it out, and Gamow and Crick and others did important theoretical work. But in 1961, biochemists Marshall Nierenberg and Heinrich Mattei cracked the first piece of the code. And over the 1960s, other biochemists worked to figure out the rest, including how RNA works. Also in 1953, University of Chicago chemist Stanley Miller and his advisor Harold Urey produced amino acids, the building blocks of life, out of an electrified broth of not living nutrients. The Miller-Urey experiment supported the idea that all life on Earth arose in a primordial soup of basic nutrients billions of years ago. Some scientists, though, including Crick, found this unlikely and thought life on Earth probably came from outer space, an idea called panspermia. The discoveries of 1953 marked a new era in biology. Evolution now had a molecular basis. Mutations are copy errors in DNA, rare but inevitable. Mutations give rise to the variation that Darwin and Wallace described. Molecular techniques revolutionized the study of evolution. Species were regrouped by the similarity of their DNA, not their visible physical features. Crabs, for example, evolved several times millions of years apart. It turns out that having armor-like skin and claw hands and being able to digest literal trash is super useful in all kinds of different watery environments. Another use of the newly deciphered genetic code was industrial. Arguably, biotechnology had been around for a while. Beer, after all, is made using engineered strains of brewer's yeast, but this process takes a long time and involves strain selection, or picking types of yeast with useful properties, not molecular scale editing. After 1953, scientists started looking for genes connected to traits of interest. The problem was knowing what genes code for what traits wasn't useful without having a way to move those genes around. So biotech took off in the early 1970s in San Francisco after Paul Berg, Stanley Cohen, and Herbert Boyer published the results of experiments with recombinant DNA, or rDNA, new synthetic sections of DNA made by cloning sections from one organism's genome into another. With rDNA, scientists could splice sequences of DNA. Berg became the first person to join DNA from two different species in one microbe. rDNA allowed scientists to copy the genes involved in the creation of the very important hormone insulin, which regulates how much sugar the body has in its bloodstream, into bacteria and yeast. Before rDNA, people with diabetes had to get insulin from pigs or other animals, but synthetic insulin is more pure. Industrial genetic engineering exploded. In 1980, the Supreme Court of the United States heard a landmark case called Diamond v. Chakrabarty. The question was whether or not a company could patent a bioengineered life form, a microbe designed to eat up spilled oil. SCOTUS said yes, if you engineer an organism's genome, then it becomes a technology. And by 1980, the biotech industry also had its first initial public offerings, or IPOs, where shares of the company become available on a stock market. Several companies launched with massive valuations. And universities, especially around San Francisco and Boston, began to view their scientific discoveries as major sources of money. They set up offices of technology transfer or licensing. Scientific knowledge and life itself became potential technologies. Next time, we're going to look at how biological technologies changed medicine and agriculture. It's time for the birth of big pharma, GMOs, and IVF. Crash Course History of Science is filmed in the Dr. Cheryl C. Kinney studio in Missoula, Montana, and it's made with the help of all these nice people, and our animation team is Thought Cafe.
All right, so that gives us a good idea of how biotechnology developed over the years. It really hasn't been around that long. 1980, I was alive. I remember 1980 well. So again, it's fairly recently that we have come up with the techniques to do some of these types. All right. So basically, biotechnology is any type of technologies developed using a living thing. And generally, it's microorganisms that we're able to manipulate. That doesn't mean that's the only thing. We're going to talk about genetically modified crops and animals and things like that later on. That's also biotechnology, right? But we utilize these microbes more than we do larger organisms because we know the genome and it's easier to tweak that genome than it is in a regular organism. Eukaryotic organisms, much more difficult to do some of the gene editing, all right? So recombinant DNA technology, where we inserted modified genes to produce what we want, it's called genetic engineering. And again, this took off in bacteria. And then from that knowledge, we were able to apply that to eukaryotic organisms. So today we have all kinds of genetically modified crops and animals and things that we use for food sources. So far, so good. Things look to be fairly safe. But again, it hasn't been around that long. So, we may see some things down the road that we don't expect right now. Yes, I got your message. I couldn't respond. If I did, it cut the movie off. I was going to message you back, but yeah, you're fine. All right. So what we have to do with recombinant DNA is take a vector, and we need something that we can use to move that DNA to another organism, right? So we need some kind of self-replicating DNA molecule that we can transport from one organism to another. And that's what a vector means. A vector is just a means by moving things. We talk about disease vectors all the time, like mosquitoes or a disease vector. They take it from one organism to another. So if you get malaria, for instance, that mosquito picked up that parasite from something else and then give it to you. And so it's a vector. So that's kind of what we're doing here. We take a gene from one organism and then we put it into another one, and then we clone that organism. We basically make copy after copy. And that's another reason why It's much easier to do this in bacteria because they naturally clone themselves. They naturally go through binary fission and they replicate their DNA. And once we've got that DNA there, then all the rest of the organisms are going to have that from then on, right? Now, the one drawback to bacteria and viruses as well is that they can escape very easily, right? So if we modify one of these organisms and it gets in the environment, we don't know what it can do, and they're so small, it's easy for that to happen. They're even thinking now that maybe COVID was even, left the lab where it was modified, and it was modified to make it more contagious to humans. So again, we got to worry about some of the ethics of this stuff and make sure that some of these organisms don't get out in the environment. I read a good book about, oh God, it's been at least 15 years ago, about genetically modified crops and how they got out into the environment, and it was a whole big disaster, and caused all kinds of weird mutations and things to occur. It was fiction, but it still made you think about these things, that some things can get out that we don't need out there, especially in the environment. Crops, we worry about that because what if the genetically modified crop takes over, and then all those genes get passed to the normal crops, and then everything gets those genes, and then something comes along that can kill that crop. We may not have corn, for instance, anymore. It'll get wiped out. That would be a big deal because we get a lot of fuel and food, and animal food especially, from corn. So we have to be really careful with some of this technology. Right, so the typical procedure for modifying, we get a vector such as a plasmid, we take that out, and then we alter that plasmid, put it into a transformed bacteria, and then we basically make copy after copy after copy of it, and then that produces what we want, all right? So again, we use vectors to move these things around. So we take a plasmid, insert a piece of DNA in it, and then we use that as a vector to go into a new bacteria that's transformed, and then that can, create a new gene that we can harvest and utilize to make a certain product, or we can make a plant resistant to certain pests. We'll see that later on. We'll talk about some of that. But again, we just basically take a gene, insert it, and then that gene now will be expressed, and then we get the product from it, right? Now, tools of biotechnology. We have done this for centuries. Selection is just by selecting for a naturally occurring microbe that does something we want it to do. Just like you mentioned with beer, over the years, beer we've made, but we've modified yeast by selecting certain ones and then growing those and keeping those, so we can make the certain flavor that we want. Bread, same thing. We're going to see that in a video in a minute from San Francisco, the sourdough bread that's created there. Right? We've done this with all kinds of animals. Just think of your dog. We have selected for everything from a Chihuahua to a Great Dane, and we as humans made those animals. We didn't make them by going through and changing their genes, but we bred them to get those certain traits. And we've done that with our farm animals. So again, it's one of those things that we've done over the centuries, is to make things better by breeding the two cows that produce the most milk, or breeding the two biggest cows to get the most meat. So we've done that naturally by selection. Now, mutations, remember we talked about these in the last chapter. If we have mutagens that can cause mutations, then we can sometimes use that to get a desirable product. So we can actually use a mutagen to mutate an organism and then get a desired product out of it. And then site-directed mutagenesis is where we target that and just basically target a certain gene, enhance it, and get a desired product that way. Right? So again, you're going to see a lot more of this in your lifetime, because the genie's out of the bottle now. With CRISPR and other things, we're going to be able to modify almost everything. I would be willing to bet that you will see designer babies in your lifetime if you're in your 20s or 30s, right? It's just going to happen. People are going to be able to select exactly what they want and then create a baby that looks exactly like the parents want. It sounds creepy, but that technology is there, right? we'll be able to do that with animals. Select a certain dog that you want. If you want a dog that's pure white or you want a dog that has black spots, you can breed for that and not necessarily breed for it, but you can actually go in and tweak the genes and make sure you get that. Right. We used to raise English bulldogs. Well, my uncle did, and I'd breed with them. But white was the desired one because Uga was white. And so everybody wanted that white dog. Well, it was really hard to breed for a white dog. You got lucky because there's so many genes involved, you couldn't predict it. So we would have several litters and maybe get one white dog out of several litters. And then the other dogs, yes, they would sell and they went to good homes, but they didn't sell for as much. If you had a pure white one, it would go for $4,000 or $5,000, whereas a brown and white one might go for $1,500, right? So if we come up with this technology, we'll be able to create anything we want. The problem with that, though, if you're a dog breeder, is if that technology's there, everybody can do it. So they're not rare anymore. So the price goes down. Right. So again, there's trade-offs with technology when we do things like that. All right, so this is going to go through sourdough, and he's going to show you how they keep producing the same flavors of sourdough by preserving the yeast that makes it. And it's a pretty interesting process here. There's one place in San Francisco that has used the same dough for well over 100 years. They just keep adding a little bit of the old dough into the new dough, and it re-inoculates it. And they just keep making the same bread over and over again, so they know it will come out the same every single time. And it's patented, it's theirs. Nobody else has that yeast that will give that particular flavor. So this is going to go over almost like a storehouse for all of these different types of yeast, and they store them in these containers, and then when they want to make a certain type of bread, they'll pull it out. But it's pretty interesting. So let me cut my mic and
in order to try to get as many sourdoughs as possible identified in the world, I am on a quest for sourdough. For the moment, we host 107 starters from more or less 20 countries. And so we ended up with 17 sourdoughs, 17 bakeries in Italy, 40, 41, 42 from Hungary. The 64 was an old lady in China. It's sourdoughs with a story where I know the people behind. Like 79 is from Josie Baker in San Francisco. 104, 106, 107 go back to the Klondike Gold Rush. Every library has a librarian. Well, it happens that in this sourdough library, I'm the sourdough librarian. When an idea like a sourdough library pops up and you are able to do it from scratch, well, it's my baby. That's why I grew so much into it. With this one, I have a very special relationship because it was the very first sourdough starter we had. This was my very first sourdough I saw in my life, and so I started refreshing it, and I had to bake with it. I immediately got interested by this dough that I'd never seen before. And being a baker, you want to know more about what's going on in there. And then you start feeding it and you see it bubbling and it's alive. It's like a pet. In the 90s, many people had this little Tamagotchi, the little thing that you had to feed. And now if you killed it, it was okay because you could just reset the whole thing and start all over. Well, here not. We have living sourdoughs. They are bubbly. Every 2 months, we open the jars and we feed the sourdough with their original flour, following the original recipe, and that guarantees that they are not changing. The fact that we decided to create this library is first of all to preserve its biodiversity. If tomorrow someone who has a sourdough that maybe he or she inherited from its father or grandmother or whatever, and if he or she would stop cultivating that sourdough, well, it's lost. In English, we use the word sourdough, but in many cultures, there is no link to sourness. The French ones, they say levain. the Italians, they use lievito naturale, which means natural yeast. But the most beautiful description for a sourdough is the Spanish word masa madre, which is the mother dough. When a daughter in the past was going to get married, she received a piece of the mother dough with her in order to feed her family. Sourdough is back on the map. Today, there is a revival. Many people rediscover again sourdough, and that's the beauty. Sourdough bread is so much better for humankind than just regular bread that is fermented with yeast. I think that every bread has something good, but sourdough just brings it to another level.
Right. So from there, you can see how certain things are selected for. So again, this is the old type of biotechnology where we utilize living things to make things for us. Today, again, we are able to modify genes and we're able to go in and actually change the genome. Instead of selecting for certain traits, we go in and create them. All right. Now, what led us to the modern way of doing things is what's called a restriction enzyme. And these are naturally occurring in bacteria, but they basically cut DNA in a specific spot where we know where it's at. And we know it's going to cut here and here. So then we can go in and splice another piece of DNA in that will match up to that area that's been cut. And that's how we insert these. So it creates blunt ends, two ends cut in the same place on both strands, or staggered cuts called sticky ends. And then the DNA can come and match it back up with that. So what we have to do is we take a restriction enzyme, we hit a plasmid, for instance, and then we take, let's say, E. coli DNA, and we cut that. It's going to cut at the exact same spot in the E. coli as it will in the plasmid that we're going to modify. And then we can just take that little snippet out, put it in, and it automatically matches right back up because the sequences are the same on the ends of it, right? And the best way to look at that is in kind of a diagram like this. So we take our target gene and we cut the specific areas, and then we take our plasmid and cut the same area. Notice that these two ends match up. So AATT will match up here, and then over here, AA and TT will also match up on the other side. So that restriction enzyme is specific for this area. Notice it cuts the same thing here in the plasmid. And so now we can go insert that target gene in, and that gene then will start expressing itself, and then it'll create a new protein, such as insulin, for instance. Right? So these are just some of the enzyme restriction enzymes that we get from various bacteria. And you can see that it has a recognition sequence where it cuts at a specific place. So this one cuts between G and G. This one cuts between G and A. This one cuts between G and C. And this one cuts between A and A. So we know when we use this enzyme, that's exactly where it's going to go in and cut that DNA so that we can cut another piece of DNA with the same enzyme that will match right up to it and fit right in. Right? So again, it seems kind of simple now that you look at it, but being able to do this was a huge leap in technology. Being able to harvest these enzymes and then utilize them. Right, so again, we take a restriction enzyme here. We have the GA site that's going to be cut, and the G and A site here is going to be cut. So we take and pull this piece of DNA out, then we can insert this DNA from another source, comes in, and then we can take ligase to put this back together into a plasmid. And now we have, now we have the other gene inserted in there, and it will start, excuse me, producing whatever protein that it codes for. All right. Again, it seems pretty simple, just cut and paste, but that's really essentially what we're doing. And we're cutting and pasting DNA instead of words, like in a Word document. Now, again, vectors are going to carry new DNA to desired cell, and these must be able to self-replicate. So they have to be a piece of DNA that will replicate on its own. So plasmids are a good sections of DNA to use, or we can use viruses. Viruses are really good at replicating once they get into a cell. So a virus, we can put a piece of DNA in a virus. The virus then goes to a bacteria, infects that bacteria, sticks that DNA in, and then it starts to replicate. Right? And there's other things called shuttle vectors that exist in different species, and they can move things between organisms. So you can even go between plants, animals, fungi, and bacteria. The other thing with vectors, they have to be stable, which means they can not break down. DNA is fairly fragile, so typically we have to have it encased in something. So a plasmid is a good way to do it, and a phage is a good way, which is a virus. And they're stable enough to where they can carry that DNA without it being damaged to the new cell. All right. So if we have a plasmid used for cloning, if we know the origin of replication that allows for self-replication, we can go insert our gene here. That way we know it's going to replicate once it gets in here. And then we have marker genes, and we'll look at the ampicillin resistance and the LAC-Z in a little while. And we'll look at something where there's a color change associated with those when we grow bacteria, so that we know we could put the section of DNA in there, right? So if it's inserted, then we can tell that clone or that colony has that new piece of DNA. If it doesn't change colors, we know it doesn't. That way we can select for the right one. And that's how we tell that the vectors actually put the DNA into the new organism. So this is going to go over recombinant DNA and show you how this is done with restriction enzymes and how it actually inserts the DNA into the new cell.
This is Bob. Bob dreams of being a world-renowned soccer player. He wants to play in the biggest leagues and in the biggest games, but there's a major issue stopping him from attaining this goal, and that's his rare and severe case of hemophilia. He bleeds a lot from everywhere. His skin is very sensitive, and so he gets wounds often. He experiences pain and swelling in his joints, mostly in his knees, elbows, and ankles, and when he goes to the bathroom, there's more blood to lose. So let's get into exactly what causes these symptoms. This is a DNA molecule, and its genes are responsible for giving us our characteristics. Their genes are responsible for making proteins called clotting factors. The clotting factors are what help us clot blood and stop wounds from bleeding. Unfortunately for Bob, the gene for a certain type of clotting factor called factor 7 is not working properly, and that's why he experiences these symptoms. Now let's see the effect of this within the body. Here we have a blood vessel, and of course it has plasma and red blood cells inside. The blood is flowing normally, but let's say you get a wound. There's going to be a tear in the blood vessel, making it lose a lot of blood. Now what usually happens is the clotting factors will stop the blood, helping in the sealing of the tear. But for Bob, because they're not working properly and they're not as abundant, they'll not be able to stop the bleeding. But how do we fix all of this? The answer is recombinant DNA technology, which refers to a series of procedures used to join together DNA segments from different species, and you'll see what two DNA segments we combine later on. In treating Bob's case of hemophilia, we're going to use recombinant DNA technology to produce a bunch of clotting factors. And this is done by taking the factor 7 gene and having it replicate inside bacteria. We do this using a plasmid vector. A plasmid is a circular DNA molecule that exists outside the bacteria's main DNA. It can safely carry the clotting factor gene into the bacteria and have it replicate. The plasmid also contains genes that give the bacteria special advantages, and one of these advantages is antibiotic resistance. This plasmid in particular is a PBR322 plasmid. It has a tetracycline-resistant gene and an ampicillin-resistant gene. So the first step for getting the factor 7 gene to replicate is to insert it into the plasmid. This is done using restriction enzymes. They cleave in the plasmid at locations called restriction sites, which contain specific DNA sequences recognized by the restriction enzymes. Each restriction enzyme recognizes a specific sequence, and so when it comes across it, it will cut it from the plasmid. In this case, we're using a restriction enzyme, EcoR1. Now that the EcoR1 restriction enzyme has cut at its restriction site, we can insert the factor 7 gene in the plasmid. This is now a recombinant DNA molecule, since we have combined DNA segments from two sources, the the plasmid from the bacteria, and the clotting factor gene. The second step is to get the plasmid into the bacteria, which is done by bathing the plasmids in the bacteria in a calcium chloride solution, which makes it easier for the bacteria to take up the plasmids. Let's take a closer look at what exactly happens in the solution. As you can see, the bacteria and the plasmids are submerged, and our goal is to get the plasmids into the bacteria. Most of the bacteria actually won't take up the plasmids. It might be that only one or two take up the plasmids, so that's why we put in many copies. And even worse, we can't actually see which bacteria took up the plasmids and which didn't. In order to separate the ones that did take up the plasmid and the ones that didn't, we have to culture them in antibiotics. Why? Well, remember, the plasmid can give special advantages, such as antibiotic resistance. The plasmid we used had resistance to the antibiotic ampicillin. Only the bacteria that took up the plasmid containing the ampicillin-resistant gene would survive. Slowly, each bacterium without the plasmid would die. Once we have the bacterium with the plasmid, we can replicate it. And those bacteria can make the clotting factors, since they all have the factor 7 gene in their plasmid. After they make the clotting factors, we extract them and mix them with sterile water so they can be injected. Now, we can give the injection to Bob and he'll feel much better. Maybe, just maybe, he could chase his soccer dreams after all.
All right, so that was kind of a simplified version of recombinant DNA technology. So again, basically you take a restriction enzyme, you cut a snippet out, put a new piece in, seal it back up, get it into a bacteria, grow that bacteria on a media that contains an antibiotic. Since the plasmid has an antibiotic resistant gene, that's the only bacteria that's going to survive, is the one that actually pulled that plasmid in. It makes colonies, you pull that out, and then you grow those. And from then on, you have a bacteria that has that gene, in this case, that makes the clotting factor 7, right? We do the same thing with insulin and other things like growth hormone, right? So again, it seems pretty simple, but figuring out that technology was the hard part. Now, the polymerase chain reaction, this is where we amplify DNA, right? And if we need to use DNA for analyzing something, we can't just use one molecule of DNA and actually work with it. There's just not enough of it. So we need to make that small quantity into large quantities. And we do that through PCR, or polymerase chain reaction. So one gene can produce billions of copies in a few hours. And then we can use these for diagnostic tests, for genetic diseases, detecting pathogens that are hard to culture, so that we know what somebody has, for instance. So if you go in and you have some kind of bacterial infection and they can't culture it from your blood, then they can run it through PCR and then look at the DNA and figure out what it is you actually have. This is only possible because of a bacteria called Thermus aquaticus, and it was found in Yellowstone in some of the hot springs. And it has an enzyme that can work at a really high temperature, and that high temperature is needed in order to carry out this reaction. So basically, we take reverse transcriptase, and then we take mRNA as a template, and we just copy it over and over and over and over again until we get quantities that are large enough to use. So if we look at what we have here, we have DNA, and so we separate the strands at high temperature. So we get this up to 94 Celsius, not quite the boiling point, and that will denature the DNA. It will make it break in half. And then we put in a primer, nucleotides, and DNA polymerase, and then we're going to come in and make a copy of that. All right. So once we copy that, then we copy it again and again and again, and we cycle it over and over again until we get many, many copies. All right. So basically, we just take and replicate it over and over and over again, and eventually we get tons of DNA that all have the same exact code. Now we have a usable quantity that we can see. We can carry out electrophoresis and see exactly what the DNA is. We can match it up to something else. So let's say you have a bacteria that we can't figure out, we can't culture it. We can run its DNA through PCR, run it through gel electrophoresis, and then we can compare it to known profiles for specific bacteria and then figure out which one you have from that. Same thing happens at a crime scene when they look for DNA from blood. Well, blood doesn't have a lot of DNA, right? So they have to amplify it through PCR. Then they run the electrophoresis gel, and then they can compare it to a suspect. If it's a match, it's a match. Same thing with paternity tests, right? You give them a swab, they amplify the DNA, they run it through electrophoresis, compare it, and see if there's enough base pairs that match to say that you are the father. So this is going to take you through PCR and just show you how this works so that you understand how we keep making copy after copy of DNA.
PCR is a powerful technique that allows us to make billions of copies of specific pieces of DNA. The first step in PCR is to open up the two strands of DNA to expose the genetic information encoded by the nitrogen bases, A, C, T, and G. In the next step, custom-made DNA molecules help us find the beginning and end of the DNA piece of interest among the billions of letters in the genome. These genetic bookmarks are called primers. The third and final step is to fill in the genetic information between the primers. An enzyme called DNA polymerase adds the building blocks of DNA called nucleotides to create an identical DNA strand. DNA polymerase works in our own cells to replicate our own DNA each time a cell divides. The first step in PCR is known as denaturation and is enabled by high heat. The second step is called annealing, and different primers have different and very specific annealing temperatures, typically between 50 and 60 degrees Celsius. The third step is called extension. DNA polymerase used in PCR works best at 72 degrees Celsius. In these three steps, we've taken our original DNA molecule and obtained an identical copy of a very specific fragment. If we repeat these three steps one more time, we'll go from 2 copies to 4. Repeat it again and we'll obtain 8 copies. And again, and we'll get 16, and 32, and 64, 128. Well, you get the idea. DNA amplification by PCR is an exponential process. After 30 copy cycles, we will have 2 to the 30 or 1 billion identical DNA copies. This is enough DNA, for example, to detect a pathogen in blood such as HIV or Ebola, or to amplify trace amounts of DNA found at a crime scene, or to enable any of the other powerful applications of PCR.
Right, so you can see how PCR reaction works. Again, it's fairly simple to think of, but coming up with that science to begin with was very complicated. But again, due to Thermus aquaticus and the high temperature that its enzymes can work at, we are able to figure that out. And we're now able to make copy after copy fairly easily. We could even probably get one in our lab and do it. I know when I was in college, we used to do PCR all the time, and that was back in the 90s. So we have the technology now to have small little machines that can do this, relatively inexpensive. So if you were in a university setting or something, you would probably be able to do PCR. All right, whoops. So how do we get foreign DNA into cells? Well, we can go through simple transformation. If you remember, bacteria, or at least some bacteria, have the ability to pick up DNA from even a dead cell and incorporate that into their own. And that's kind of what you saw in that first video when we were talking about, going through with restriction enzymes and, making recombinant DNA. We just allow it to sit there, and hopefully a couple of them would pick up some of the DNA, and then we were able to target which ones did by seeing which ones were ampicillin resistant. The other thing we can do is electroporation, where we make an electric current around the cell, and that kind of puts tiny little holes in the cell, and that allows the DNA then to move into the cell, and then that gets incorporated into its DNA, and that becomes part of it as well. So again, we can use these techniques to get that DNA into that cell so that we can transform it, or basically make recombinant DNA. And another thing is protoplast fusion. We can remove the cell wall from 2 bacteria cells, and then we can allow those to fuse together, and then those segments will fuse together as well. And now we have a new chromosome that has both DNA from one cell and the other. So again, this would be our recombinant cell down here. Another way is a gene gun, and it's about what it sounds like. We shoot DNA-coated bullets into a cell. So you'll take it, coat the DNA on a bullet, shoot it in. It's not like a bullet like from a gun, but it's tiny, and we can shoot that into the cell, or we can do micro injection where we can put foreign DNA into an egg. So if we wanted to clone an animal, for instance, we would do the micro injection. You take an egg cell, remove the DNA from that egg cell, and then place new DNA in there, reduce that egg to start to divide, and then we end up with a cloned animal from that. Now, genomic libraries, this is very similar to what we saw with the yeast library that they had earlier. They basically kept a collection of different types of yeast. Well, in this case, we collect different clones containing different DNA fragments. And we can take and digest and splice these little snippets of DNA into a plasmid, put those in a bacteria, and then we can hold that so that when we need it, we can go to the library and get it. Now, some of these are very expensive. So if I wanted to order a specific gene to do some research with, I could go to this company that has a genetic library, and I could get whatever gene I wanted. And then I could use that gene and then transform another bacteria with it and get that to do what I wanted it to do. Right? So they try to get at least one clone for each gene in the organism. That way they have an entire library and they can pick and choose which gene that they need. Right, so let's say this is our genetic material, and these are genes, the red, the orange, and the green. So we would take restriction enzymes, and then we would cut this up, and then we would put the red gene in one bacteria, the orange gene in another, and then the green gene in another, and then we could store those, and then we would always have bacteria with that particular gene in it. And that way we could go pull it out, reconstitute it, and then get that dividing, and then we can make copies of that gene. Same thing with a phage. This is a bacteriophage or a bacterial virus, one that attacks bacteria, and we can store that gene there. And then if we ever need to use that gene, we can infect a bacteria with the virus. It'll insert that gene, and then that bacteria will start expressing that gene. Right, so we basically take complementary DNA, and then we make that from messenger RNA using reverse transcriptase, which is the enzyme that will create that. And we do this with eukaryotic genes because eukaryotic DNA has introns that do not code for a protein. So we want those out of there. And those get removed when we go from DNA into messenger RNA. So then we can take that messenger RNA and then reverse that process and make a copy of that into DNA. And then that will essentially give us a true gene that doesn't have the introns involved. So you can see that in this diagram. Up here, we have exons, which are the things that we want. And then we have introns, which are the kind of junk DNA that we don't want. And so when we make RNA copy, it initially has a copy of the intron, but these are typically removed. There's enzymes in the nucleus that remove those and make a pure form of messenger RNA. And then we take reverse transcriptase and then we run that backwards, and then we make an exact copy of DNA from that. And now we have DNA without the introns in it. Notice how it's all purple down here. And then the mRNA is digested and removed. So now we have the DNA that we want, and that way we can store that for later use. Right, so this is a DNA synthesis machine. So again, this is going to carry out those reactions we just looked at. Again, it's fairly complicated, but the machines today do most of the work for us. Again, most of this stuff is very repetitive in labs now. So once you turn the machine on, you learn how to operate it, it does all the work for you, and will make these copies. So you don't have to physically, as a person, go in and do it. Now, originally, someone had to come up with a way of doing that and build a machine. But today, we're so far along that these machines do most of this stuff for us. All right, so blue-white screening. This is similar to what we looked at earlier in the video about recombinant DNA. But we use a plasmid vector containing ampicillin resistance and a B-glycosidase gene. And the bacteria is grown in a media containing ampicillin and X-gal, which is the substrate for B-galactosidase. And then only bacteria that picked up the plasma will grow because the ampicillin is there. They have to be resistant in order to grow. And the only way they can be resistant is if they picked up that plasmid. Then only the ones that have the new gene will not hydrolyze XGAL and they appear white. And then the blue colonies that did not pick up the new gene will appear blue. And that's how we separate these out. So that you know which one did pick it up and which one didn't, so that the one that picked it up, it can be selected and then cloned and copied from there. So this kind of shows you how that works. So again, we have a restriction site here. We put our foreign DNA in, but this plasmid contains ampicillin resistance, and we grow it on ampicillin, and the only ones that can grow have to have that gene, and then the only ones that turn white are going to be the ones that contain the foreign DNA. So we can guarantee that this plasmid picked up that new piece of foreign DNA because, one, it picked up the plasmid and it has resistance because of that. But two, now that it turns white, we know that it picked up the actual foreign DNA as well. So it's kind of a double-step process here with this one. So again, we know it took up the plasmid because it can grow on ampicillin because it's resistant. And then two, we know that it picked up the foreign gene because it's white on the plate. And the blue ones we can discard because they didn't pick up that piece of DNA. So again, that's the way we can separate these things out. All right, so I'm going to stop here and then we'll pick up next time with your biotechnology projects. So make sure you get those in tonight. Again, those are due at midnight. So if you haven't turned that in already, make sure that you do. If you have to do something at the last minute, get it done. But we'll start watching those Thursday. I will make a link to it so I can just pull them up and we can watch them together. All right. I'll try to grade those tomorrow so that I already know what's in each one. So when I select them, I already know kind of what's, what the topic was and that kind of thing, so that I can explain some stuff to you as we look through those. All right. So that's kind of where we're at. We're kind of getting towards the end of the class where we're mainly focusing on projects. So we only have really one more chapter after this one to cover. I believe that's chapter 14. That'll be on your last test. So we'll cover it later on, but we'll start watching projects and doing that kind of thing. Remember, with your disease project, you have to make up your questions and get those submitted as well. So I can go through those and compile them so I can give you a list of those questions. What you're going to do while you're watching each video is answer those questions, and then I will put those exact questions on your final test. So you will know half of your final test before you even start taking it. Right? All you have to do is memorize all those questions, but you're going to have to watch each video to be able to answer those questions. All right. So again, when you make my copy, give me the answer, but then make another copy that doesn't have the answer that I can give the students. And make sure you follow the format correctly. Times New Roman, 12-point fonts. That way I can just cut and paste, and it doesn't take me a long time to go through all of those. Okay? Now, does anybody have any questions? We'll finish these slides up probably the following week on Tuesday, so I can finish up. There's not a whole lot left in this chapter. It's fairly short. So we'll pick up here next time. So does anybody have any questions before we head out? Not today. We'll have one when we finish the chapter. That's why I kind of went up to the last minute. I usually give you a few minutes if we're going to have one. Well, chapter 21 through 26 is going to be covered by your disease projects. So basically, you're covering what's in there. So we're going to learn about all that stuff as we go through your project. So I'm not going to go and lecture. It would be the most boring lecture you ever saw, me showing you every bacteria that exists, and they all look alike, and you'll cover the main ones that you're going to see in the field. And that's the main thing, is to know the disease-causing bacteria that are out there so that you can recognize them when you start work. Right? Yes, 5 questions, multiple choice. It's all in the instructions. Just make sure you follow those to the letter. I am going to grade this one a little more strict. All right. Nothing on the next test. You'll have to memorize all this stuff. Oh, the test that's coming up. Yeah, that one you can use metabolism drawing. Yeah, the one due Sunday. Yeah, you can use those. Any of the handouts that I gave you online. Yes, we will have class calling. We'll be watching your projects. Do you remember which one that was? I'm trying to remember. If I gave it to you, yes. I'm sure it was important if I told you to print it out. Just make sure you hold those up to the camera when you take your test. I can see what you got. I'll go ahead and move that test out now so I don't forget about it. Yeah, definitely use that one. Yeah. That one's actually going to be more beneficial than some of the other ones. Yep. Again, just hold them up to the screen. I'll put the instructions on there.